Hello class, today we will be doing the IR experiment. The purpose of today's lab is to use IR instrumentation and interpretation to determine the identity of an unknown compound. The entirety of the IR experiment will be performed on the IR instrument. The unknown compounds are in separate vials. The cleaning solvents isopropanol and methanol will be used in between the compounds to clean up the IR instrument stage using CHEM wipes. Before we jump into today's lab, let's go over some general lab safety. Here comes a student that is almost ready for lab. She is wearing pants without holes in them and shoes that are closed toe. However, her hair is not tied back and she is wearing a long-sleeved sweatshirt that is not flame retardant or graded for lab chemicals. On her second try, the student is now ready for lab. She ties up her hair with hair ties so that it is out of her face. She puts on her lab safety goggles and she puts on her lab coat, which is fire retardant and is graded for lab chemicals. The student also double checks her list of chemicals for today's lab and puts on her lab gloves. Let's review some proper lab attire. You must be wearing shoes with closed toe and no height. This means no heels, no platforms, no Crocs, no peep toes, and no sandals. You must be wearing pants or floor-length skirts that do not have any holes. Short sleeves are preferred. If you have long hair, it must be tied back. If you have a long beard, it must be tied up. In every lab, you are required to use the proper PPE, or personal protective equipment, gloves, and goggles. Now let's go over the lab safety stations. The emergency eye wash and safety showers can be found in one of the front corners of your lab. If you get a chemical in your eye, go immediately to the eye wash station, pull down the eye wash station door. The water will start running immediately. Keep your eyes in the stream of the water for 15 minutes. If chemicals are spilled on your body or your clothing, immediately go to the safety shower, remove all of your clothes, pull down the safety shower handle, and throw the curtain around you. In the other front corner of the lab is where you can find the fire extinguisher. The fire extinguisher should only be used if you are confident in its use, the fire is small, or you are using it to escape the fire. Here is where you can also find the glass waste and spill kit. In the event of a spilled chemical or broken glassware, please inform your TA. Your TA will be responsible for cleaning up the mess. Now let's get into the specifics of the IR lab safety. Today we will be using a series of unknown compounds. When you work with unknown compounds, you have to assume multiple hazards associated with them so that you can perform the experiment as safe as possible. In this case, we want to make sure that we are wearing our proper PPE, gloves, and goggles. We know that we are using isopropanol and methanol. The hazards associated with isopropanol and methanol is that they are toxic and flammable. Because they are toxic, we want to make sure we do not ingest or inhale them. Because they are flammable, we want to keep them away from spark or flame, such as electrical outlets. We begin today's experiment by going to the IR Instruments computer and loading the software Omnic 9. Next, we need to make sure the IR stage and crystal are clean by spraying it with isopropanol or methanol and wiping down with a CHEM wipe. Now that the stage and crystal are clean, we can take the background of the instrument. Because we are measuring the spectra of volatile liquids, we will take the background with the volatiles cap in place over the crystal. Here is a straight-on view of the computer's desktop. Open up the Omnic 9 software in the bottom left-hand corner. We close the first pop-up that comes up and maximize the screen. Next, we open up the experiment setup in the top left-hand corner. The first thing we want to make sure is that we have 8 scans selected in the number of scans. The final format of the spectra needs to be in percent transmittance, so we make sure it says percent transmittance, save, and then press OK. Now that the experiment setup settings have been saved, we need to collect the background of the instrument, so we click on Collect Background next to the Experiment Setup button. We press OK and wait for the 8 scans to be performed. Around scan 4, we can see that the background is starting to appear. When the scans finish, it asks you Add to a new window, press No. Now that we've taken the background of the instrument, you might be wondering what is happening inside the instrument. This is a block diagram of the Michelson interferometer inside our IR instrument. A block diagram shows the relationships of the parts of a machine, but not necessarily the exact locations or dimensions. An interferometer is a device used to measure the interference pattern of electromagnetic waves. The Michelson interferometer in our IR instrument starts with the IR source emitting the IR radiation. The IR radiation is split into 2 directions by the beam splitter made of potassium bromide. One beam is directed to a fixed mirror, which is reflected back towards the beam splitter. The other beam is directed towards a moving mirror. The moving mirror moves back and forth in the same direction of the wave, causing this beam's phase to change with time. As the beams recombine at the beam splitter, they combine in a specific pattern of interference, and this new beam is directed towards the detector to be measured. Before the IR beam can reach the detector, it passes through where the sample is located. Here's a diagram of an older model of our IR machine. Instead of a sample compartment like in the diagram, we have the sample stage. Our stage is diamond crystal that is IR transparent, meaning it does not absorb IR radiation. As we can see from the diagram, the IR radiation passes through where the sample is placed before reaching the detector. When we have no sample placed on the stage, the IR radiation will interact with the atmosphere in between the stage and the sidearm. The interference pattern created by the interferometer, in addition to the interaction of the atmosphere, gives us our background spectra by the detector. Now that the background is done, we take the volatiles cap off of the stage. We obtain a vial of unknown sample, unplug the dropper, and carefully place 1 drop of the unknown liquid onto the crystal. We carefully replace the volatiles cap onto the spot, making sure not to touch the sides of the cap to the liquid, and we can take the spectrum of the sample. Now that the sample is loaded, we can take the spectra. Next to the Collect Background button is the Collect Sample button. We click on Collect Sample and the pop-up will ask us to enter the spectra title. For the spectra title, we type in the unknown number and press OK. The second pop-up reminds you to load the sample, which we have done, so we press OK and wait for the 8 scans to be complete. When the scans are complete, it asks if we want to add to a new window, and we click Yes. Now that we have our spectra, we need to find our peaks. Click on the Find Peaks button, which is next to the Collect Sample button. It will automatically place the horizontal line in the middle of the spectra. Click at different heights on the screen to see how the horizontal line finds the peaks. Click towards the top of the spectra below the baseline to find all of the peaks. Now we know the specific wave number for each peak, and we can print our spectra to include in our report. Now that we have our spectra, we need to clean the IR stage and crystal. Remove the volatiles cap and spray either isopropanol or methanol on the stage and wipe with a CHEM wipe. Wipe the stage dry with a new CHEM wipe. Now that we have collected the background and sample, how does the IR produce the spectra? The interferometer produces an interferogram for the background and the sample. The instrument converts these interferograms to their respective transmittance spectra by performing the Fourier transform of the interferograms. The Fourier transform is a type of multivariable calculus. In this case, our variables are p, path length difference in the interferometer, and v, the frequency of the wave or wave number. Once the Fourier transform is complete and the background and sample spectra are produced, the instrument subtracts the background spectrum from the collected sample spectrum to produce the final spectrum of the sample. Now that we have the spectrum, how do we interpret the spectrum? First, we need to know what is happening on a molecular level. Molecules are continuously moving. Their vibrational energy, or the oscillation frequency of their bonds movements, are within the infrared region of the electromagnetic spectrum. There are two aspects of the molecular bond that we need to consider. First is the mass of the atoms at either end of the bond. Lighter atoms, such as hydrogen, move much faster than heavier atoms. Therefore, bonds involving hydrogen oscillate faster, which means they have a higher vibrational frequency. Second is the bond order. Of the bond orders, triple bonds have the shortest bond length, which means that their oscillations are shorter and therefore more frequent, so they have a higher frequency, followed by double bonds and then single bonds. So both the mass of the atoms in the bond and the order of the bond help influence what the peak's wave number will be. Now we have to consider how the atoms in the molecule are moving. There are two main types of bond vibration, stretching and bending. Stretching changes the bond lengths of the molecule. Symmetric stretching happens when adjacent atoms are moving in the same direction along the line of their bonds. Asymmetric stretching happens when the adjacent atoms are moving in opposite directions along the line of their bonds. Bending vibration changes the bond angles of the molecule and thus do not occur along the lines of the bonds. Molecules can bend in the plane of the bond in two ways, scissoring and rocking. Scissoring occurs when adjacent atoms are moving closer and further from each other, and rocking when adjacent pairs of atoms are moving closer and further from each other. Molecules can also bend out of plane by wagging, where all adjacent atoms are moving in the same direction out of the plane of their bonds. These types of bending and stretching are called vibrational modes. The stretching vibrational modes are higher in energy than the bending vibrational modes. To sum up, the vibrational mode, in addition to the mass of the atoms and the order of the bond, determine the frequency or wave number of the bond's vibration. Now that we know how the molecules are moving, what is happening at the IR instrument at the sample stage? At the stage, the frequencies from the interferometer interact with the vibrational frequencies of the molecule. When the frequencies of the bond vibration and interferometer are the same, they couple together in what is called constructive interference. This is what creates the peaks that we see. The intensities of the peak are determined by the amplitude of the wave. Bonds that have a large dipole or a high difference of electronegativity along the bonds will have higher amplitude waves, which will produce longer, more intense peaks. Conversely, bonds that have small dipoles will have shorter, less intense peaks. Now that we know what is happening in the machine and on a molecular level, let's dive into how we interpret the IR spectra. The primary use of IR spectroscopy is to identify what functional groups are present in the sample molecule. The IR spectrum can be broken down into 4 different regions of characteristic stretching vibrations. The highest frequency bonds are the atoms bonded to hydrogen, followed by the bond orders, triple bonds having the next highest frequency, then double bonds, and ending with the single bonds in the fingerprint region of the IR spectrum. These are the 10 compounds being analyzed in the IR Spectroscopy Lab. By looking at the molecular structures, what functional groups do we expect to see in our IR spectra? We have different carbon skeletons, alkanes, alkenes, and aromatics. We have some oxygen-containing compounds, alcohols, aldehydes, and ketones, and we have one nitrogen-containing compound with a nitrile group. Let's go through some examples of what the expected peaks look like for these functional groups. Alkanes, the most basic hydrocarbon, have 2 types of bonds, carbon-hydrogen bonds and carbon-carbon bonds. As we learned earlier, the carbon-hydrogen bonds will have the highest frequency because the hydrogen atom is the lightest atom. The carbon-hydrogen bond also has a high difference in electronegativity, so we are expecting the peak to be strong. The carbon-carbon bond contains atoms that are equal in mass and electronegativity, so the peak that we expect to see is lower in frequency and amplitude. Alkenes, or compounds with a carbon-carbon double bond, will have a peak that is higher in frequency than a carbon-carbon single bond. As we learned earlier, the bond length of the double bond is shorter than the single bond, which means the frequency is increased. If the carbons of the double bond have other alkyl groups attached to them, the peak of the double bond becomes shorter with each substitution. This means that something is happening to the electronegativity of that bond. As you increase the substitutions around the double bond, the electron density of the double bond is becoming more dispersed. This is because the empty pi orbitals of the carbon substitutions attract some of the electron density of the double bond. When you have a double bond connected to mostly hydrogens, the electron density of the bond is concentrated in the double bond because the hydrogens do not have pi orbitals. So the peak is a little bit stronger. When the double bond has hydrogens on it, you will also therefore have a CH bond. As we know, CH bonds are the highest in frequency. The CH bond off of an alkene is even higher due to the hydrogen being more attracted to the pi bond of the double bond. However, this also means that the amplitude of the peak will also be shorter than the alkyl CH bond. Aromatic groups are similar to alkenes due to having several double bonds. However, in an aromatic ring, the electrons are delocalized. This means that the electron density is not concentrated at each double bond, but instead it flows around the ring. The bond order is actually 1.5, which is in between a double bond and a single bond. This means that the carbon-carbon bond of the aromatic ring is also in between the frequencies of the double and single bonds. Because of the pi character of the bond, we also get a weaker peak that is closer to the double bond order as well. The amplitude of these peaks are weak to medium because there is an electron flow despite the equal electronegativities of the atoms. Similar to the alkenes, the level and type of substitutions will also affect the amplitude of the peak. Also similar to alkenes, aromatic CH groups also show up higher in frequency than the alkyl CH groups because the bond order of the carbon is higher. The carbon-hydrogen amplitude is usually weak due to the magnetic field created by the flowing aromatic electrons. Alcohols are the easiest group to spot. They have a higher difference in mass than carbon-hydrogen or nitrogen-hydrogen, so they are the highest in frequency. Due to the hydrogen bonding property of alcohols, The peak that we see is broad. The electronegativity difference in the oxygen with two lone pairs and hydrogen is large, so the peak is also usually strong. A lesser important peak you will see is the carbon-oxygen bond in the fingerprint region. It is lower in frequency than the carbon-carbon bond because oxygen is heavier than carbon. Because of the difference in electronegativity between carbon and oxygen, the peak will also be medium to strong. Another group that is easy to spot is the carbonyl group. A carbonyl group is a carbon with a double bond to an oxygen. The carbonyl bond is higher in frequency than the alkene bond. You might be thinking, but oxygen is heavier than carbon, so why is the carbonyl bond higher in frequency? It is higher in frequency because in the carbonyl bond, the carbon is partially positive and the oxygen is partially negative. This means that there is an attraction between the oxygen and carbon that makes the bond stronger. The stronger bond means a higher frequency. Following the same logic, due to the difference in partial charge and electronegativities of the atoms, carbonyl peaks are also strong. Aldehydes contain a carbonyl group that is attached to a hydrogen. Because the carbonyl carbon is partially positive, the carbon-hydrogen bond is weakened. This makes the carbon-hydrogen bond of the carbonyl lower in frequency and amplitude. Also unique to aldehydes, the carbon-hydrogen bond peak can be split into two peaks by a phenomenon called Fermi resonance. The peak can be split into two when the stretching mode and bending mode of this bond are close in frequency. Ketones are almost the same as aldehydes, except we do not have a CH bond on the carbonyl carbon. Instead, we have two alkyl groups attached to the carbonyl. Because alkyl groups are electron donating, they reduce the positivity of the carbonyl carbon, which makes the carbonyl bond a little weaker, making it a little lower in frequency than the aldehyde carbonyl. However, the ketone carbonyl will still have a strong peak due to the difference in electronegativities and partial charges of the atoms. Finally, our last functional group of our unknown compounds is the nitrile group. The nitrile group has a triple bond, which is the highest bond order. Therefore, the nitrile group will be higher in frequency than the alkenes and carbonyls. This peak will also be strong due to the electronegativity difference of the carbon and nitrogen.